Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 72, and it's going live on August the 21st. This week, you could not make up the fact that 5 straight days of rain has coincided with an electrical fault in my car, meaning the windows won't go up. Still, it's now nice and clean inside and out, and I can wash my feet as I drive. School is back here, so the house is very quiet, and I can't say that I like it too much. And that's about it for what's happening. I'm sure I'll remember something amusing or interesting that happened this week as soon as I finish recording. I do think the summer vacations are drawing to a close because the inbox is growing again. Most of it is irrelevant though. I've also had lots of bank invoices that have been paid by companies I've never heard of to a range of banks across the world that I don't have accounts with. I suspect it's probably spam. Last time we had a conversation with JJP Biologics about specially built antibodies to tackle autoimmune diseases and cancer. This week it was remarkable that the interview worked so well given we had 3 guests and 2 other people watching, and me. The screen looked like the Celebrity Squares show. I don't think that still exists in the UK, but I think it's called Hollywood Squares in the US, and it might still be running as far as I'm aware. What this has to do with biotech, I have no idea. I sidetrack very easily. Oh yes, there were lots of people on the call. We chatted about the Flexible Automation for Scalable Health program, also known as FLASH, and it is focused on the development of scalable cell-free DNA bioproduction capabilities. So, our guests are John Scheel, Program Manager at ARPA-H, DNA Script co-founder and CTO Xavier Gaudron, and John Nelson, Senior Biosciences Principal and Flash Program Lead at GE Healthcare's Healthcare Technology and Innovation Center. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, or sponsoring a podcast episode, or for event coverage, or maybe just to tell me your vehicle horror stories so I don't feel so bad. So first up this week, it's a repeat guest, which doesn't happen too often, so it was nice to renew acquaintances and talk about Utah again with Sethera Therapeutics CEO Karsten Eastman to get some updates and to chat about the announcement of a collaborative breast cancer research project with Roswell Park Comprehensive Cancer Center. Hi again, Karsten. Good to have you with us for the second time. And maybe before we get to all the latest news, for those that didn't hear the first conversation, could you maybe give us some background on the company?
Yeah, absolutely. Sethera is a biotechnology company that's focused on developing a new type of peptide medicines that we call polymacrocyclic peptides. Peptides sit in this space between small molecules and the larger biologic drugs like antibodies. They can interact with proteins in ways that small molecules often can't, but peptides have historically had some limitations around things like stability, delivery, and the types of structures that can be made. What we've done at Sethera is make an enzyme, and we're an enzymatic technology that lets us staple and change the structures of peptides in different ways. And importantly, we can make multiple connections in the same molecule. And that gives us a lot of diversity that was previously inaccessible. And our goal is to go after important targets that have been difficult or even impossible to address with the drug technologies that we've had before.
What kind of success have you had with that so far?
One of the pieces that has been really interesting is some of the peptides that we've pulled out of our screens have had longer time on say, the disease target than even antibodies. So this has been one of the nice validations is that we are getting antibody-like properties, but with much smaller sizes.
Is AI making a difference in that process?
What's great is we're actually upstream of AI. So all of the architectures that we're generating have never existed before in any database or have never been screened at the scales that we're screening, which means that we are generating the necessary data to make some of these future AI models better. But one nice piece is because we have multiple staples in our peptides, it actually makes some of the computational optimization a lot easier to do. And that's where AI and ML can be an important factor.
You mentioned the polymacrocyclic peptides. Could you tell me a bit more about the platform?
Sure, at the core of it, we use an enzyme or multiple enzymes to create very strong bonds at specific positions within the peptide. These bonds can either constrain the peptide into one ring, several interconnected rings, or even higher order architectures. And that's where the term poly, many macrocyclic rings, many rings, peptide comes from. And one way to think about it is a normal peptide is kind of like a little string where it can adopt a lot of different shapes. But if you put specific connections in that string, like, you pin it in a location, you can pre-organize it into the shapes that you want. And we don't just make one type of shape. We've really expanded the diversity and the architectures. One thing in nature that's really important is structure equals function, and we've tried to expand and generate as many structures as we can.
You mentioned structure and function. Is there always function? I mean, are you generating things that have potentially no uses?
Of course, think of it also partially like an alphabet. There are some words that just don't mean anything. And it's a similar idea with peptides. A peptide is a small protein, and nature has spent so much time evolving the proteins that are in your body, that are in other areas of the environments. And peptides can be small proteins or small words that have no meaning. But this is where a nice analogy is we're taking the dictionary of everything that could have function and the dictionary of everything that might have function and another dictionary that has no function and throwing all of them at the same time at the targets. But that's where we can find the right word for the right disease state.
And I guess what might not be a word in English probably is in Welsh. Does that apply though, that there are some peptides that have uses in, or you think are going to have no uses, but they do in an entirely different capacity?
Yeah, another good way is consider individual biology. So my biology is different from yours, and your biology is different from other people that are living both around you or far away from you. And the way that you brought up language is that there are some peptide words or peptide sequences and structures that could affect me, but would have no effect on you, and vice versa. So it is very much the case that individual biology can respond, and this is even true with small molecules and biologics. The individual biology can respond differently.
So it's kind of like on a personalized medicine level in that respect.
Yes, and I think peptides can address personalized medicine in some very powerful ways.
What can these peptides do that hasn't been possible previously?
Well, I want to maybe think about this as there's something called chemical space, and what chemical space is are just all of the different chemistries that could exist. And for a long time, the way that we've done drug development has been very limited in that chemical space. We have this tiny slice that we can access. And what peptides are doing, or these our technology and other technologies in the peptide space, is that they are opening up the systemic exploration of larger chemical space. So this basically means that we can make these molecules that have never been screened before. And because they've never been screened before, there's a lot of opportunities to address disease states that have been previously considered to be undruggable. We're not assuming before that we're going to know what the perfect shape is for this biology, but we're just, throwing so much at it that we then pull out what is appropriate. So that's really what it is. We just have more access to what's possible.
And then what's the way that you work? Is it through partnerships that the partner utilizes those peptides, or how does that work?
We're doing two different routes right now where we're developing our own internal assets where there's biology that we have a lot of interest in. And at the same time, we are looking for active partnerships where they have a biology of interest that they have been trying to work on for a long time. And small molecules maybe haven't worked, traditional peptides haven't worked, biologics haven't worked, and they want to try something that is new and different and has a lot of enablement associated with it. So that's two different routes. We have our own, and we also want to partner.
All right. Speaking of which, could you tell me about the mutant P53 breast cancer program and the award?
Of course. So this was a congressionally directed medical research program that we applied to. It was a very competitive process. Less than 4.5% of the applications that successfully went in were funded, and we are very excited about this program. P53 is often called the guardian of the genome. In healthy cells, what it does, it can, I guess, recognize when something goes wrong and can prevent those damaged cells from continuing to grow. It's a little more complicated than that, but that's the idea in a nutshell. But P53 is also one of the most commonly mutated proteins in cancer. Certain mutations change its shape, and this is again structure equals function, and interfere with that protective function. We have a collaboration with Roswell Park Comprehensive Cancer Center in Buffalo, New York, and received this award. And it's really interesting because this mutant P53 is fundamentally a structure problem. And instead of actually trying to, say, inhibit a protein, what we're doing is we're discovering peptides that can change the abnormal form of P53 back to its protective normal state. And it's a really difficult biological problem, but this is a type of problem that our platform can address.
And what does that mean in terms of what it will mean for patients?
What it will mean for patients is if you have, say, a very difficult to treat type of cancer and it isn't responding well to normal chemotherapy or these other possibilities, this is a way of basically resetting that protective protein back to its normal state. And then the cancer itself will undergo an apoptotic or a death process without the need of any harmful chemotherapy agents. This is using the body's natural processes that have been turned abnormal by the mutations and resetting it back to a normal functional state.
And obviously, you're not at this stage yet where you're doing this, but what kind of delivery would that involve?
Here, I think that it would still be safest to assume that it would need to be an injectable based on we want to ensure that the right amount of peptide is getting to the cancer. There are opportunities for oral development of peptides, but that's a very separate question of does the peptide get to the right place? And because we're dealing with, in this case, very difficult to treat cancers, here I think that we're still too premature to say that it would be orally bioavailable.
And so, how is the project with Roswell Park Cancer Center going to work?
I think it's really very complementary. Our role at Sethera is to discover and characterize those polymacrocyclic peptides. And then the Roswell Park team has great biology and really deep expertise in P53 and cancer models. And the question that we're really asking is not can we make something that binds P53? That is an interesting question, but we want to know if these are altering the p53 in a biologically meaningful way. So Sethera is set up to do the screens, and then Roswell Park is set up to actually look at what is being done at the biological level.
Is it going to involve clinical trials at some point?
There would need to be more development. The goal is to move the peptides that come from this towards clinical applications. So, because we have this partnership with Roswell and we have some great people there working on it with us, if we see the right responses in animal models, we will move this towards clinical trials.
All right. Is there any time scale on that?
So the CDMRP is set up to run for 3 years. I think that we can get a lot done during those 3 years, and after that, start moving it towards the clinical applications.
And obviously, breast cancer is something that a lot of companies are looking at. How do you see the future of the battle against breast cancer?
Well, there's great reasons for optimism right now. And the way that I've looked at it is we're moving away from thinking about breast cancer as a single disease. Cancer is incredibly complex, and two people can have breast cancer, or really any cancer, and the drivers behind those tumors can be very different. This is one of the reasons why, solving cancer has been such a problem. And, you and I were talking earlier briefly about some individualized medicine, and the future is going to involve increasingly precise treatments based on what are the vulnerabilities of that individual patient's cancer. And there are ways that we can look at this by the different types of sequencing that we can do and looking at, okay, how is this being driven? And we've already seen great advancements from certain types of targeted therapies, things like antibody drug conjugates, where you can have an antibody that is bispecific, goes after two different areas in the cancer with a great conjugate to it that will help eliminate it. And I think that the next frontier is expanding what we can actually target and what is the modality that is doing the targeting. And I think that peptides have a great future there.
Do you see with on that personalized medicine front, do you see that being more single therapies or a whole range of different therapies specifically tailored to individuals in terms of the amounts and the ratios?
What I see in the future are ways of taking already validated therapeutic approaches that work well for, say, set A of cancer cells. And when we're, say, looking at an individual's cancer, they might have, say, some of set A, some of set B, and some of set C. And if we understand what molecules have worked well in that space, we may be able to combine the therapies appropriately for that individual that are targeting A, B, and C simultaneously. Now, whether that's individual compounds that are mixed together to address that, or if it's a single compound that has different moieties that can go after, say, three things at once, that would be a trispecific. That's how I see it being addressed. We would first understand, okay, what is the exact type of cancer and the mechanism that you have in that cancer? And then how do we take the therapies that have worked well in these individual places and combine them for a better effect?
And you mentioned the way that your company's working, and also you're working on this project with Roswell Park. What else do you have in the pipeline or that you're working on concurrently with this?
One area that we are also excited about is the inflammatory pathways. We've seen that the peptides have had just fantastic success with the GLP-1s, so semaglutide, Wegovy, tirzepatide, all of these molecules in the weight loss space and diabetes treatment have been immensely successful. And, I understand and respect that's great. A certain population needs that. But the other side of this is, I think every person experiences inflammation, and as we get older, inflammation becomes a larger and larger concern. And, NSAIDs work to a certain amount, steroids work to a certain amount, but there are specific inflammatory pathways that just appear to be perfect for peptides to block and modulate. And that's another area we're currently working on.
Well, hopefully we will have Karsten on again when they have some more good news. Next, we're going to talk about AI and drug discovery with Valo Health Chief Scientific Officer Karin Conde-Knape. Valo Health works on drug discovery and development through its AI-enabled human causal biology and closed-loop chemistry platforms, and it has its own pipeline. Hi, Karin. Great to have you on the podcast today. I wonder if maybe first you could tell us a bit about the company.
Yes, Valo is a company that was created around 2019 with the intention to transform the way that we drug discovery. And by that, I mean that we are starting by putting the patient at the center first and then ending with the patient. So utilizing AI technology and as an enabler to help us understand the patient trajectory from a health status to a disease. And in that context, being able to understand what are molecular and biological drivers that are responsible for their trajectories, with the understanding that not every patient is the same. And therefore, the way that we are used to treat, it's no longer viable. And so understanding the patient, coming up with the biology, and then putting on top of that a platform for AI-driven predictive chemistry that allows us to get to the best molecules possible also in shorter periods of time. So an end-to-end platform that connects the human centricity with the chemical approach to get to drugs, better drugs for the right patient.
Now, you've had quite the background in the biotech field. I wonder if you could just go into a little bit of that and what you're doing now at Valo Health, because you just moved there, not that recently, but maybe why you chose to go to a smaller company.
Yeah, so I have spent more than 20 years in the pharma industry, in big pharma and multiple pharmas, always in the early phases of target ID towards early clinical development. And I have always been very curious as to how are we enabling better patient understanding if we are really serious about bringing differentiated treatments. And during my tenure at Novo Nordisk, I was lucky to come across Valo Health as a company that was precisely intending and as a mission having this connection of understanding of the patient and then delivering better treatments. And that hooked me since the beginning. Not only because, it's not only the way that you talk about AI, it's about applying it to the right data sets with the right scientific questions. So when the time came for me to change roles and departure Novo, this was an opportunity that came. And I knew the company. I had established a collaboration in Novo with Valo during my tenure. And I knew already the team, excellent quality team, scientifically and so. And it was really a great opportunity to now come and join as the Chief Scientific Officer. So, I get to do what I truly believe in, which is, again, this understanding the patient to deliver better treatments, and with a scientific process and an approach that you can extrapolate across different therapeutic areas, but also very importantly, that you do not need to silo yourself in one, but let biology guide you in the many areas where biology is overlapping, and that you can really make the best out of a potential target or molecule across indications rather than thinking in a siloed way, which usually happens in bigger corporations.
Obviously, size is one difference. Are there any other differences that you've noticed now that you're working at a smaller company?
Absolutely. I mean, I have been a key driver of innovation via the external ecosystem while I was at my previous companies. And of course, what is, biotech and the dilemmas that they suffer, but it's very different when you are actually living it. The conversations are different. The decisions that you make actually matter. There is no place to hide. You are having direct influence on the science and decisions and bigger decisions for the company. And that on one end is really refreshing, but on the other hand, why not? It also can be scary, right? So it's being able to be closer to where things are happening and then see the dynamics in a smaller corporation where things matter and the partnerships that you have matter. Those dilemmas and interactions are actually quite important and impactful. So it is very different, but it's fun as well, and it's refreshing.
And you get to know everybody's name, I guess, which you don't at a big company.
Yeah, absolutely. Absolutely. I had very large global organizations in the past, and yes, it's very difficult, right? And here that's also a pleasure. You get to spend a lot more time closer to the science and where things are happening, and to see firsthand really the excitement, the passion, the novelty that is coming directly from the scientist, almost on a day-to-day basis. And that makes my day. At the end, I'm still a scientist at core. So that is also very, very fun to be able to see the new pieces of data as they come fresh.
One of the things that we've noticed is there's been a big increase in the number of AI providing companies, and they all do something slightly different. But could you tell me how Valo Health is different to the rest?
Yes, I think one of the important elements that we have as a company is the platform that connects, as I said, the human central element to the chemistry. So it's not only because we are accessing human data. I mean, many companies are accessing human data, but the difference here is that we're able to access not only very broad, very deep data cohorts. We have about 20 million data pieces with the very deep phenotyping of the patient through electronic health records, for example, and that is coupled with omics, such as genetics or other sorts of omics. And we are able to triangulate these different elements to let the data paint the patient. So rather than just relying on what the code is telling you about the disease, we are letting all the different parameters tell you what the patient really has. And through that tool, we are able to really convert this knowledge of the patient into what are the molecular drivers and then apply the chemistry. And from a chemistry perspective, we are front loading all the optimization of the parameters that make a drug in the very early stages of chemistry design. So rather than spending time of optimizing potency, for example, and then later on finding that you have to optimize PK or that you optimize solubility, we are doing all of that very front-loaded. And in addition to this, we are utilizing virtual and DNA-encoded library screens in very, very large data sets in places or with chemical representation of the molecules in a way that we do not need to have starting points. We do not need to just optimize, better molecules. We do not need crystal structures in order for us to discover chemistry. And I think that allows us to go into targets, not only that are novel, but where we are not relying or dependent on existing chemical matter for us to be able to just optimize. So when you have that combination, we are able to bring targets that hopefully will have better probability of success. We are in a way running the human experiment much earlier than even doing the first wet lab experiment because of the triangulation that we are doing for selection of the targets. And therefore, the hope still needs to be demonstrated, of course, is that the targets that we are working on in a pipeline that is very diverse will have better probability of success.
And I guess AI has started to be used in a lot of companies for a while now. How do you see AI being utilized in the drug discovery process?
Yeah, it's interesting that, of course, everybody talks about AI, and in some instances, it is seen or perceived as the solution to all our problems. And the main problem that we have in the pharma or biotech side, it's that 90% of the programs that we have for drug discovery and development fail. And this is something that we are all trying to fix. The challenge is that you do not have a tool, no matter how good the methodologies are becoming, that will really help you solve these. And in a few months, you'll have the solution from target ID all the way to the market. So AI, it's an enabler that it's starting to mature in certain areas. For example, in many instances, when you have a known piece of biology or a validated target, because there is already a molecule in the market, you can utilize an AI as an enabler to generate better chemical matter fast. And that is possible because it's known biology, you have all the tools, and you are applying your algorithms to come up with better molecules. And that has been an area that has matured significantly, and where I believe it's AI as an enabler is making good progress. On the other hand, when we're talking about the novel targets, so brand new, where you don't necessarily understand what is the biology or how well the connection of the target with the driver of the disease is, and therefore tools do not exist, you do not have chemical matter, you don't have really a path that is set to just run and prosecute your project. This is where it becomes a little bit more challenging. Right, so you do need to utilize methodologies helped with AI, first and foremost, to help you digest all the amount of clinical data and human data that exists. And then hopefully through other enablers are the existing data in the in vitro setup that hopefully together will give you that path towards validation and prosecution. This is still an area that is more in maturation, but it's still, I think, on the early days, and therefore, again, not the full connection that you can see. In other areas where AI, I think, is also being very, very useful, it's from the utilization of large language models to help you redact regulatory documents, to help you also analyze certain images from tissues, samples, or from any other cellular readouts that you have, where it helps you see patterns that you may not be aware that you can utilize or that are actually correlated to functionality, for example. So in buckets and pieces, there are definitely areas of help where the tools are enabling the scientists to make better decisions and maybe uncover patterns that are actually relevant to the disease and to the function that were not known before. However, as I said, there is still a long way to go before we are able to just press a button and then come up with a drug, which I think is where everybody would like, and that is just not going to happen anytime soon.
Yeah, definitely. Do you think that the way that it's being used needs to be changed?
I think we need to be very much aware as to where it can help, where it's really an enabler, where are the caveats, where are the assumptions that need to be, tweaked, and then also be very much cognizant that a model is only as good as the data that you utilize to train it. So if the data that you have available for training a model is limited, the pattern recognition or the amount of the opportunity for the model to learn is going to be limited. So access to the data is also in some instances limited, and not because there is no data, it's just that the quality of the data is not right. So I think more than changing the way that we use the tool, I think is more, are we clear in the questions that we are asking the tool to solve? And to be honest, I think this is where we are struggling the most. It's not that we don't have access to algorithms. It's not that we do not have access to data. It's just that we have not really been able to frame the problem in the best way so that the answer that we get out of the tool is actually helping us. if you put a team of computer scientists together with a data set, they will spend a lifetime analyzing the data set and providing you with output of this. But if the initial question is not framed in the right way, the output is going to be there, but not necessarily impactful. And I do think that for this to be more impactful, we have to be more strict and cognizant as to what is really the question that we are posing.
I think in a lot of cases, we tend to hear more about and concentrate more on drug discovery, how AI can help speed up drug discovery and how it can impact drug discovery. But does it also have a role to play, and is it being used efficiently in this respect? Things like analysis of results, not to find out whether it's working, but also reasons why it isn't working?
Yes, I think in some of the questions that we have been able to frame is try to understand, when you have a molecule that maybe did not deliver the efficacy that you were expecting, you are able to go back and really look at the more parameters that maybe you had anticipated to utilize. Here again, coming back to electronic health records or coming back to in clinical trials where you had very much defined your parameters that are primary and secondary endpoints, and therefore your analysis focuses there. But when you're able to integrate the totality of the data and then ask a different type of question, then it can start to help you discern why this worked or didn't. I will give you an example. When we are developing drugs for weight loss, usually the tendency is to think that weight loss equals improvement in all the different comorbidities associated with weight loss. And while the pure weight loss effect might actually be enough to drive some of these improvement in comorbidities, the reality is that it's not necessarily only the weight loss that it's the one responsible. So by doing this type of mediation analysis, utilizing some of these tools, you are able to start to dissect effects that are actually happening even before you have a significant amount of weight loss, or you can start to understand that weight loss is not the only responsible for the effect that you're seeing, or the lack of effect as well. So by using these type of methodologies, you are able now to start to understand much better, well, first, is the mechanism that you think you have the only one responsible? Through the integration of omic data, are you able to then dissect what additional mechanisms are at play? And even important also is if you are interested in bringing different type of treatments, can those mechanisms become the next target that you are able to bring forward in a drug discovery process? So by all means, I think this is another approach in terms of learning the type of trials that you are running, the type of population that you are using. You can also start to simulate trials by utilizing these AI methodologies, which, if you think about it, the amount of time and savings that you could have if you are able to do these experiments in silico rather than to spend, millions and, 2 or 3 years in the clinic for you to figure out that maybe you were selecting the wrong population, which is a very painful exercise and very costly as well.
Do you think that as well as the drug discovery process, that AI has a role in helping with changing the diagnostics field?
Absolutely. So when I talk about understanding the patient, it's precisely, that we stop looking at the patient on the basis of a code. And I'll just give you a brief example, because we are at the moment utilizing these methodologies of integration of these very massive data sets to help us paint subgroups of patients. So we have done this, for example, in heart failure. And when you are letting all the electronic health records of, thousands of individuals in a longitudinal fashion, you have many traits that you can incorporate and then start to segment really a patient. And then you realize that if you ignore codes And then you let the data paint, really you start to see a different segmentation. So a patient that maybe you had diagnosed as heart failure, you start to realize that this patient has many other comorbidities, or that in this other patient, there are different biological drivers of a heart failure that are not necessarily the same as others. So from a diagnosis perspective, I don't believe that in the near future, it will be enough to say, heart failure as the only label that you have, but you are able to go maybe one or two levels underneath to decide to say, yes, this is this type of heart failure, and this is maybe some of the drivers that are contributing to the progression of the disease in this population or in this individual versus somebody that is completely different. So through this approach, you will also be able to come up with tools, biomarkers in this case, that will allow you to identify the population or the patient and not just know that we are talking about a heterogeneous disease, but how do we find them so that we can actually treat them. So it is going to start changing the way that we are diagnosing and the way that we are providing treatment. And this is all part of the new, I think, drug discovery and development approaches, rather than keep seeing everybody at the same, and not only at the same, but even with wrong diagnosis sometimes.
How important are partnerships between small and large biotech companies, and also the relationships between them and AI providers and academia?
Yeah, I think this is actually quite fundamental in the ecosystem that we are operating, because not a single group, and this you can call it big pharma, biotech, or academia, is able to solve the problems that we have at hand. Doesn't matter what is the indication that you are talking about. The reality is that there is more to be learned about it, and more to understand from maybe more precision medicine approach. Both pharma and biotech need each other on one end, because pharma has the big muscle to be able to carry forward, ideas and clinical trials in multiple indications potentially at the same time, while biotech has the nimbleness to move faster and to maybe be a little bit more risk tolerant, to try and test approaches that in a larger organization would have much more difficulty to do. On the other hand, academia also has, in many instances, the innovation and the know-how on diseases, but they lack the know-how of drug discovery and development. And therefore, this is where you start to see synergies that when you put them together, hopefully you are able to enable a path much faster and in more relevant way towards the delivery of a new treatment. So I think it's fundamental. I think it is it's something that needs to continue to be tailored as well as work through it, because you were asking also dynamics. There are very different dynamics when you are working in your big corporation, your small, but in the interface, it's also quite important. It's very important to have a collaborative approach here, because again, it's a synergy, and the synergy, for this to work, you really need to have the true element of collaboration and capitalize on the value that each of the partners bring to the table so that the magic can happen. And this is not always easy, I must say. It's not always easy to find that right partnership, because sometimes there are different agendas and there are different understandings of what is that each of the partners is contributing. And that's when things can go a little bit wrong. But always through collaboration and communication, I think history has said it, right? Many of the big wins have come from this type of partnerships in the past.
Are you able to tell me anything about some of the partnerships that you have at the company and some of the successes?
Yeah, we have a couple of partnerships. One with Novo Nordisk, that, as I said, I was behind setting it up while sitting at Novo. This is a partnership that has captured two different disease areas, one in cardiovascular and one in obesity and diabetes. These are partnerships that the main element is target discovery and validation. And they have been quite successful in helping to frame the thought process as to how do we go about utilizing the big and very, very large data sets. What are the specific research hypotheses that we are putting on the table to deliver, the next set of targets? And while I cannot go into the specifics of the research hypothesis, what I would say is that through this collaboration, we have been able to put elements in place that now can be replicated and then utilized in other disease areas, right? So we have been able to go from a specific research hypothesis to a more of a population view now back into a research hypothesis, which allows us to do maybe exploration in other new therapy areas. So out of these collaborations, we have several projects in validation and in drug discovery pipeline. And the second collaboration that we have is with Merck Serono, and this is in Parkinson's disease. And this is also very exciting because it's also about delivering new targets, but very importantly, is helping us define how is the disease progression. So we are very excited about this. This is still in early days, but it's also one where we are expecting to deliver a number of targets into the Merck pipeline.
And obviously, AI never stands still. As it evolves, how do you see the evolution of drug discovery and its use in the biotech industries?
I think this is a tool that is here to stay. Not tapping into that in different shapes and forms in drug discovery, I think it's a mistake because it's a wagon that is ongoing. And if you don't jump into that wagon, you are risking to be left very way behind. And as I said, as there are still areas of maturation and areas of improvement, certainly it's a matter of being more risk tolerant and being able to have fast iterative cycles. Because the proof of the pudding of that, the methodology, the tools are delivering better quality targets and with higher probability of success is not going to be demonstrated until you are in the clinic or even so that you are able to bring this to the market. And in between, it's all the different proof points that you need to have to demonstrate that indeed you are in the right path, right? That you are selecting the right target, what is the population that you're gonna go after? Do you have the right tools to enable? So all of these is gonna continue to happen. And the devil is in the detail as to the specific methodologies that the different teams are using, as you said. And I think the more complete path that you have through the tools helping you make better decisions, the better you're going to be set. So we need to keep an eye on it because it's going to become a reality sooner rather than later, more than it is now. And it's how are we using it and what are the questions that we're asking about it.
How do you, as a company, adapt to that change at Valo?
So we are in a position where, through the experience that we have gathered over the last 6 years and plus, we need to continue maturing as well. And we need to continue building models that will help us expedite the way that we understand the validation of the target, for example, or that we prosecute that validation. But also it's not only about the target ID. I mean, target ID is just one small area if you want. But then the potential that you can have by harnessing all of that value into how you design your clinical trial, how do you position in the market? So it's across the whole value chain that you are able to utilize these very large data sets in order for you to gather and making maybe the best decisions for the molecules that you have at hand in the future. So it's an evolution and we'll continue to evolve. We continue to making sure that we are utilizing the data at hand in the best way possible to generate the most impact across the value chain. I just think that we need to keep making whatever we can in terms of efforts to better understand the patient that we're treating. We are all interested in doing this in a better way, but it starts there. It's a fundamental piece in drug discovery and development. And if we are not using the tools at hand today to do that, then everything else becomes a little bit irrelevant, right? If we think that just bringing one more of the same because it works, it's the best way to go about it, well, then we are still not bringing the most value to the patients that need treatments.
Well, that was a really interesting discussion about AI in drug discovery and diagnosis. Two very enjoyable chats today, as they always are. And so that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. And so until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.